Okay, we're going to try to get started now. Um, the room is kind of dark, and most of you probably have had lunch, so I will do my best not to disturb you. So the topic of today is image segmentation algorithms. My name is Dzung. I have a bunch of affiliations here that have nothing to do with NIH, but actually I'm part of this center, the CNRM, Center for Neuroscience and Regenerative Medicine, which is a collaboration between NIH and the Department of Defense. So even though nothing here says NIH, I actually work here at NIH. I'm in Building 10. So here's a picture of Einstein's brain. I always find Einstein's brain interesting because he was such a brilliant scientist and physicist. So there's a couple things that I found in this, interesting that I found in this article. This is back in Time magazine in 1999, where they have preserved Einstein's brain, and they've still continuing to study it just because of the person that Einstein was. And so what I found interesting was, one of the things was, here's Einstein's brain, and here's another preserved brain. And whoever is taking care of Einstein's brain needs to do a better job, because Einstein's brain's not looking so good compared to this one. I mean, it's kind of dried out looking, and it's clearly saggy. I don't think the brain is supposed to be this flat, especially compared to this one. So they need to do a better job. But the other thing that was interesting was Einstein's brain is actually missing the parietal operculum, which is a gyrus that most people have. But he doesn't have this gyrus, and as a result, his inferior parietal lobe was 15% wider than the normal person's brain. And so why is that interesting? Well, it turns out that this inferior parietal lobe is actually the seat of mathematical and visual reasoning. And so the scientists who are studying this brain made the conjecture that perhaps this structural anomaly in Einstein's brain actually led to his brilliance in science and physics. And so this brings back an old field of study called phrenology. So I don't know how many of you have heard of phrenology before, but it was a pseudoscience that was popular in the 19th century, where they basically believed, well, we know that people have brains, so if we study the shape of your head, the brain should affect the shape of your head. We study the shape of your head, we should be able to figure out what kind of person you are based on the shape of your head. So they had devices like the one shown here, which basically made measurements of different points in your head, and then they made predictions about, what are you good at, what are you not good at, what is your personality like. And so we know now that this science didn't really hold up through the ages, but in fact, the idea, the premise of it seems to be proven to be true today, because we do know that the shape of the brain is actually related to different aspects of how you act and disease processes, and is an interesting thing to study because of this. And so just a side note here, I have little links on the bottom left that's of some of my slides. This one is, I don't know if you can see it, but it's called museumofquackery.com. There's a museum that's in St. Paul, Minnesota, called the Museum of Questionable Medical Devices, where they actually have a phrenology machine, and I visited the museum while I was there one time, and it's very interesting. Although you could do 2 readings of this phrenology machine, and your results would be totally different. So I think it was like a random number generator of some sort. So we know that the size and shape of different parts of your brain might be related to disease processes and aging processes and function. So how do we study the size and shape of these regions? Well, this is where segmentation algorithms come in. So segmentation algorithms are algorithms that delineate anatomical structures or other regions of interest. And here's an example here of a very popular segmentation algorithm called FreeSurfer, where we start off with the MRI head scan. And here you can see the MRI head scan, and I'm just fading in the free surfer segmentation, which finds a lot of different parts of the brain automatically. Okay. So the objective of today is to provide an overview of segmentation algorithms like free surfer to better understand how they might be used in your research. So this is going to be pretty high level, just very introductory, trying to give you a sampling of different types of segmentation algorithms, different approaches, and tools that are actually available that you might be able to use. So the overall outline is shown here. It's just a rough outline of what I'm going to go through. First, we're going to talk about why are you here. We'll briefly go through the different challenges of segmentation, and then I'll go through really quickly a bunch of different segmentation approaches, both semi-automatic and automatic segmentation approaches. And then I'll try to provide some practical examples of, okay, once we know about these approaches, we're going to actually try to use one of these to run an actual scientific study looking at the disease or some aspect of the brain. And then I'll briefly go through some current and future directions of segmentation research. And feel free to stop me at any time. This should be informal. If you have any questions, you don't have to wait till the end. Just go ahead and stop me. So there's lots of applications for segmentation. I'm listing a bunch of them here. Volume quantification is probably the most popular one. In many studies of disease, we simply want to know what are the volumes of certain structures and how are they associated with behavioral characteristics of the person, or perhaps disease duration and other clinical aspects. Morphometrics is the study of shape, and so there's also studies of shape besides, which is sort of a little bit more complicated than looking at just the volume, sort of summarizing the characteristics, the properties of a structure based on one quantity. You can look at quantities that actually characterize the shape. Localization monitoring pathology is important, computer-aided diagnosis, treatment planning. But this is a fMRI summer course. So what does segmentation have to do with fMRI? So before everybody steps out saying this has nothing to do with fMRI, I'll try to explain that there are segmentation applications within fMRI analysis. So there's a popular functional MRI package called SPM. There are probably a lot of AFNI users here, but SPM is also popular. And what they use is they actually have a process for normalizing your brain. Usually you have an fMRI data set and you have a corresponding T1 image, and if you want to do a really good job of putting the brains into normalized space, you take the corresponding T1 image and you do the registration on that. Since it's higher resolution, has less distortion. Their process for doing that normalization is actually doing a combination of segmentation and registration at the same time. So this is just showing here that they're breaking up the tissue components as they're doing this registration process. There's an SPM toolbox called AAL, or anatomical automated labeling, which is a method for labeling different gray matter regions. And the idea there, or at least the original purpose, and it's used for many different things now, but the original purpose was mainly just to see where your activation's occurring. I want to know what's the gyrus where a particular fMRI activation is occurring. And then this last figure here is showing a result of a free surfer fMRI analysis. So here, you're performing your fMRI analysis on a manifold on the surface of the cortex. Some people believe that this is a more natural domain for fMRI analysis, since the cortex is a folded surface, so you want to smooth along the surface rather than through the volume. So in order to get this surface, of course, segmentation is required to reconstruct the cortex. And then just a final note, if you're doing resting state fMRI analysis, many times you want to do functional connectivity between regions of interest, compute functional connectivity. And these methods, AAL and free surfer, are common ways of actually defining those regions of interest. So all of these segmentation applications can be used in fMRI analysis. Some non-fMRI applications are listed here, and this is This is actually a clinical product that I'm showing on the left. It's called NeuroQuant, and it's the output from this software that's made by Cortex Labs. It's an FDA-approved product, so radiologists can actually use this if they're interested in computing volume. So it integrates with a PAC system, and then these outputs are sent back to the PAC system, and the radiologist can say, oh, look, this person's cortical gray matter is a certain volume. And then they also put out normative curves. So they'll say, oh, this person's brain structure is 15% outside of the normal value, above or below. We actually have implemented a fake version of NeuroQuant. This is you have to pay for each time you use this normally, so if people are interested in trying something like this but have a free version, you can talk to me. Some other applications is MS lesion segmentation. So in multiple sclerosis, people tend to get these lesions within the brain, particularly in white matter. So if you look at T1 and FLAIR images shown here, you can see it's hypo-intense on T1, it's hyper-intense on the FLAIR. And it's of great interest to know how large those lesions are. So this is actually used often in MS clinical trials. So if they're testing a therapy for multiple sclerosis, they want to know, is this drug actually slowing down the progression of lesions? So this is one of the outcome measures typically of a clinical trial, is to see what happens to lesion volume once they've taken the drug. And then just to show you that segmentation doesn't have to always be on a structural image, it can also be on other kinds of MR images. So this is an example of segmenting diffusion tensor images. Here, I'm not sure how clear it is, but because the white matter tracts are actually overlapping in many regions, there's a little bit of a crosshatch pattern, so some of the labels are overlapping, which is much different a lot of times from the typical structural segmentation. And this is just an animation of some of the tracks we constructed. Okay, so what are the challenges of segmentation? I can look at these images, and it seems like I can tell where the regions are. Why is this so tough? Or can I just get a radiologist to tell me whether a region is big or small? Well, it's true that the radiologists actually are quite good at telling how regions change over time or differences between regions, but quantitation is something that's much more difficult. It's very time consuming if you want to actually assign a number to the size of a structure. So you actually want to go through and each pixel and say, what is this in gray matter or white matter or CSF? So what I'm showing you here is sort of a cartoon of part of a brain focused on the cortex. And this is what we would really like MRI to look like. If we acquired an image that looked like this, the job would actually be pretty easy, I think, to do segmentation. However, they actually look like this. And I'm cheating a little bit here because I've already removed the non-brain tissue here. So in head images, you normally get dura, fat, skull outside of the brain. I've already removed that here to make the job a little bit easier, but still it's not that easy. So you have artifacts or limitations of the MR images that make the job of segmentation a little bit more challenging. So instead of this these nicely defined boundaries in this image, you have this, which is much more difficult to make a distinction between different classes. So imagine now going through each of these voxels and saying, I want to say this one is probably gray matter and this one CSF. You can see that it can be a maddening process. And that's why we tend to hire other people to do it and not do it ourselves. And there's actually companies that do it for you, so that's the great thing. Another thing that I'm showing here is that some of these images have RF inhomogeneities. So this is where the coil sensitivity is causing the images to be brighter or darker in different locations. So if you're just assuming that the intensity value of a pixel is the same throughout the image, then you'll get a not very good segmentation shown there. So this idea of limited resolution is something that we call partial volume effects in the images. Ghosting is another artifact that you can get from motion or hardware problems. And then another difficult challenge is, in the end, once you've applied your segmentation, how do you know that it's actually working correctly? And a related problem is if you know what the segmentation should be, then we should be able to use that to improve our algorithms, and that's where training data comes in. I'll be talking more about that. Okay, so I'll be reviewing a bunch of different segmentation approaches. It won't be an exhaustive list. There's probably some segmentation approaches that I might not be covering, and maybe I won't cover your favorite one, and I apologize. But hopefully I'm getting the most commonly used ones. And in general, the decision to apply one segmentation algorithm over another is domain specific. So it really depends on what you're trying to obtain in the end. And sometimes, if you just want to get volumes, you're not really interested in having a perfect reconstruction of the shape of a structure, then that might drive the type of segmentation algorithm that you use and how you evaluate the algorithm. So here's three different approaches, segmentation approaches, applied to trying to obtain segmentation of subcortical gray matter structures, and in some cases ventricles as well. And so you can see some differences between these different approaches. This is a multi-atlas approach, and you can see compared to FreeSurfer, it's a little bit less blocky, and whereas this FSL approach, this algorithm called FIRST, it's extremely smooth, and in fact the thalamus is quite a bit larger than you find in the multi-atlas approach. So certainly you're going to find differences between segmentation algorithms, and it's all going to end up being trade-offs with computation and time and what you're trying to get in the end, what your final outcome is. And here are a couple of recent segmentation algorithm review papers, if you're interested. So let's start with one of the simplest segmentation approaches that one can apply. This is called thresholding. And the idea is that if you have a scalar image, you want to create a binary partition of the image intensities. So again, here's an MRI brain image where I've already removed the skull. And so a histogram of this image would look like this. So what's a histogram? The histogram is basically an occurrence, a plot of the occurrence. So how many times a particular intensity occurs, you'll go up at the y-axis, and then the x-axis is just the different intensity values. So in this example, this is just an illustration, but the white matter peak is here corresponding to white matter, which is the brightest on this T1 weighted MR image. Gray matter is in the middle, and CSF is at the bottom. So you can imagine if you wanted to threshold this image and separate brain tissue from CSF, you might want to threshold right around this region here. And so thresholding is most often used as a semi-automatic method, but automated versions also exist. So here's an example of changing the threshold value as we're going through the same MR image. And I'm using a software package, which you can't see because of these icons, called MIPAV. So MIPAV is a software package that was developed here at NIH by CIT, led by Matt McAuliffe and others. And it's a very nice package for visualizing MR images and performing sort of the less complex segmentation operations. So thresholding is pretty easy to perform, as I'm showing here. So what you can see is that I'm leaving the grayscale intact for for regions that are below the threshold, and then turning red the regions that are above the threshold. And as I move higher, you see fewer regions in red, and as I move lower, you can see just about everything in red. Right? And I also mentioned down here that you could do thresholding with AFNI's 3D calc. It would be very easy to just say image is less than some threshold value. Another commonly used semiotic method is called region growing. And it's a technique for extracting an image region that is connected to your seed point based on some predefined criteria. So it's often used to a delineation of small structures like lesions, and typically requires some manual interaction to specify that seed point, although there's automated variations. So in this case, for example here, I've placed the seed point in the butamen, hoping to extract regions that look similar to the butamen. So what happens when I sort of vary a threshold based on the region growing criteria here is that it's similar to the threshold. You sort of, as you relax this criteria, you start to encompass the entire brain, and as you make it more stringent, you get areas very close to the butamen. And you should keep in mind that this is a 3D operation that I'm showing. So even though this putamen does not look like it's connected to the other side here in 3 dimensions, these things do become connected. Okay, so this threshold is typically governed by either the variation in the local intensities around that region or edge features, and we'll talk a little bit about edge features in a few slides. And I do mention another software package that has region growing. It's called 3D Slicer. It's a software package out of the Brigham Women's group, or the Boston group actually. MIT is also involved. And it has a lot of nice features as well. So this is a third method which is commonly used in a semi-automatic fashion. This is going to be the last one I mentioned that's semi-automatic. And this is called, these methods this class of methods is called deformable models. So they're physically motivated techniques for finding region boundaries by using closed curves or surfaces that form under the influence of internal and external forces. So the idea of a deformable model is that you're going to have a contour in 2 dimensions or a surface in 3 dimensions, and we're going to simulate movement of that contour or surface based on physics properties. So we could have external forces, so somebody pushing your contour towards a boundary that you want to find. So this is actually showing here a force field. So these are vectors. I'm not sure how well you can see it, but these are little arrows pointing towards the boundary, this little U shape that we're trying to find here. Some people call it a toilet seat, but I prefer to call it a U shape. So here we're showing an actual deformable contour, and you can see that it starts to move towards this shape, and it stops. Okay. So that's the external force that's pushing it to that boundary. But there's also internal forces. So I mentioned internal forces here. So what are these internal forces doing? Well, they're keeping this boundary to be, or this contour to be smooth. Okay, so you can see that even in regions here where the boundary is kind of pixelated, it's actually still forming a very smooth shape along this region. So you can think of this as, these internal forces as similar to a rubber band, which has elasticity. So if you've ever played around with a rubber band and you try to make a sharp corner on a rubber band, it's very difficult. So that's the same property that we're imposing on these deformable models. So as you can imagine, this can be semi-automatic or fully automatic, where you sort of draw a contour that's close to the region and then you let it evolve. So that U shape that I was showing, we was just showing this boundary. Typically our images don't look like boundaries, but you can actually extract boundary images from these images. So here I'm actually just computing an edge map based on spatial derivatives. So you can see that instead of having the different regions with different intensity values, the edge map is bright where there are edges and dark where there are homogeneous regions. So this is often used as an intermediate step for driving these deformable models. And there's two kinds that are used most often. They're either parametric deformable models or geometric deformable models, which are also called level set models. So what are the differences? Well, the parametric deformable models are represented explicitly. So if you're thinking of a contour, then it's a bunch of dots connected by lines. Or if it's a surface, it's typically some sort of mesh, a triangle mesh. And the nodes of the mesh exist in a continuous space. Okay, so they don't have to exist on the image grid. So they actually can be in between voxels. So that's an advantage of these parametric deformable models. But the disadvantage is that they can actually be computationally complex. So as you can imagine, there's a bunch of points that you need to manipulate now. And the other property, which is not necessarily a disadvantage or advantage, is that they generally have to have a fixed topology. Okay, so I'm going to talk about topology in a second, but the topology can't change in parametric deformed models, or typically do not change, whereas level set deformed models can adapt to topology. So this is an example of a level set deformed model from the software package ITK Snap. And here the user created 4 little circles within the ventricles, and the level set deformed to the ventricles and merged. So that merging process is a topology adaptation. Okay. So these level set deformable models are a little bit more flexible in terms of topology. They're numerically more stable, but they are generally defined on a voxel grid, so their resolution is more limited than these parametric models. So one of the major applications of deformable models is in reconstructing the cerebral cortex. So I'm showing you what it looks like when one of these meshes or deformable surfaces are deformed to find the cortex. So it's actually going in between the white matter surface and the gray matter surface. And you can sort of visualize this within the images of, in this case, the underlying image is a representation of gray matter, and these contours are representing the different surfaces. So, so in these applications where you're trying to find the cerebral cortex, you're modeling the surface as an elastic sheet, okay, and it's just pushing, same in that contour, you're just trying to push it towards the gray matter and find either one of the gray matter boundaries or perhaps the center of the gray matter, depending on your application. And this animation stops, so I'm going to go backwards and forwards so you can get more info. And for the cerebral cortex, topology preservation is an important property. So what I'm showing you here is a sphere which we're molding into the star shape, okay? And we're actually rotating it as well. All of these operations are topology preserving because we're not tearing anything in the surface. But if you punch a hole in the surface or you split it into 2 different pieces, now you're changing topology. So topology is this property. And the reason why we need to, or we like to preserve topology in the cortex is, one, we don't believe that the cortex has like these different pieces or holes in them. Okay, biologically, we don't believe that to be true. And the other aspect is that in order to do group analysis, we like to actually map things into the same space. And this is much easier to do if they all have a consistent topology. Okay, so that was the last slide that talks about these methods that can be used in a semi-automatic fashion. Most of the methods that I'll speak to you about now are fully automated methods, although they do have some requirements for initialization or training data. So this is a big class of methods that are popular for image segmentation, and these are called classifiers. So they're machine learning classifiers, really. And the idea is that we're going to represent your images as some sort of feature space. So you can think about, for example, if you have a T1 weighted image and a T2 weighted image, and you want to use all of that information combined to do your segmentation, you can form a 2-dimensional feature space So this would be, I think it's showing T1 on this axis and T2 on the other axis. And you can see that there's some nice separation of gray matter, white matter, and CSF in this feature space. So we want to use this information, this feature space, to find the different regions. Okay, so there's many different ways of doing that. Intensity values are actually one way of building your feature space. You can actually make this feature space of more complicated features that are derived from the original images. So you might run a filter and add another dimension to your feature space. You might look at the neighbors of each voxel and make that another dimension of your feature space. So there's lots of different ways of building your feature space. And they can be either unsupervised, supervised, or generative discriminative, and I'll talk more about that in a second. Okay, so there's this term that's popular in the segmentation literature called generative models, and so I have a little bit of math in here just to explain what a generative model is. So think about an image model where you have your observation Y, okay, and it's coming from an ideal image. So what's your ideal image? So we're going to assume that your ideal image has k different values. So if it's a brain, it's probably going to be gray matter or white matter CSF. So it's maybe 3 different values, and each of those tissues ideally would just have a constant intensity value. So that's going to be your V, which we're going to call either a mean or a centroid of mean intensity of that typical class. And then we're going to have this, this variable Z sub jk, which are indicator functions. So what pixel j, this is going to be 1 where you have CSF, for example, or for a particular class, and it's going to be 0 for the other classes. Okay, so that's your ideal image, and then we're just going to add noise to that, and that's going to be, and our observation is going to come from the sum of these 2 terms. Okay, this is a very simple image model, but this is where the term generative comes from, is that if we're assuming that your underlying image is being generated from this sort of statistical process where you have an explicit representation of what is happening, we can call it generative. And what is the importance of being generative? Well, it turns out that you can make your algorithms completely independent of training data, for example, because you've made some assumptions about the data. Okay, so we don't need training data. We don't need examples of what a good segmentation is and bad segmentation. We can just go ahead and segment our image. So generative models are typically unsupervised approaches, meaning that they don't require training data. So if you actually want to solve this model, we can put it into a maximum likelihood estimation formulation, and we can use an optimization algorithm. So there's a popular clustering algorithm called the k-means algorithm, which is actually solving exactly this model that I've shown you. Okay, so it's mainly just trying to find places where the observed value is close to this mean value, and then assigning the indicator function to be 1 in those regions. Okay, and then it alternates through those processes to find these different clusters. So most generative models most unsupervised classifiers, which don't require training data, are generative, but they don't have to be. So there are algorithms where I could actually take this function and put an exponent here, and I get these fuzzy C-means algorithms, which are not necessarily generative algorithms, but they're still unsupervised classifiers. And so what I'm showing you here is an example of an MRI image where the k-means segmentation has been applied. There's a variation that I can apply, which I can use a Markov random field prior, which is a way of regularizing or making the segmentation a little bit more smooth. So you can see there's a lot of dots, sort of isolated regions of gray matter in the middle of white matter, which we don't expect to occur in the brain. Well, if we apply a regularization, we can make the segmentation more smooth and get a nicer looking result. And then this final graph is just showing you, it's actually an animated graph, that we're fitting a bunch of Gaussians to the histogram of this image. And that's essentially the process that's being performed in these clustering algorithms. In particular, if we apply expectation maximization algorithm, we can actually fit 3 Gaussians to the distribution and end up with our segmentation. So it's worth noting that with certain kinds of algorithms, it's sometimes generating a soft segmentation rather than a hard segmentation. So the hard segmentation is what you would typically expect. For example, a k-means algorithm only generates a hard segmentation. It's saying that each voxel is either one or belongs in one class or does not belong in one class. However, we can actually use these algorithms, such as the EM algorithm, to generate a probability function at each location, which gives us a probability of whether a voxel belongs to one class or the other. So in here we have this example. We just have one class, which is the prototype intensity is very dark, and one class has a prototype that's very bright. And if we have observations shown here, it's pretty easy when the observation is dark to say, okay, this should belong in class 1. If it's bright, it should belong in class 2. But if it's sort of in the middle, we're not really sure what to do with those observations. So if you have an algorithm that computes a soft segmentation, we don't have this problem, right? So we just have gradations between 0 and 1 rather than having to make that exclusive decision. So this is basically saying, we have 2 different classes. Everyone is all different shades of gray. We shouldn't be exclusively assigning people to different classes. Okay, so we just talked about unsupervised classifiers and generative models. Probably the most popular method of segmentation right now are supervised classifiers. So these methods require training data. So I've been talking about training data. I'm finally going to explain what training data is. So supervised classifiers partition the feature space based on data with known labels. So the training data are data where you know what the segmentation already is. Okay? So for segmentations, this is typically manually segmented data. So you have to have data, some set of data that's where you know the truth already, or what you believe to be the truth, to actually help your algorithm perform its segmentation. So this is an example here showing you manually segmented data from this company, Neuromorphometrics, and they actually sell whole brain, manually labeled whole brain segmentations that you can download and then use for your algorithms. But perhaps what's not as widely known is a subset of this data is actually available for free, and because it was used as part of a challenge. So at Vanderbilt, they organized a segmentation challenge where they provided the training data, and they have a subset of this available for download. So this is a very useful data set right here if you are if you don't want to pay for your training data. So this is showing you a 2-dimensional feature space here, and the red and blue we're pretending that those are training data from 2 different classes. So we have a blue class, a red class, and then if we get some sort of voxel that we want to label, and it lies in this part of the feature space, we need to make a determination based on that training data of what class it should belong to, the red class or the blue class. So you can imagine now many different scenarios of how you might want to make this assignment. So, for example, I might want to just look at, okay, what are my neighbors? What are the training data that are close to me in the feature space? And I would think that I would want to be the same label as the neighbors that are close to me. Or we could actually have a more parametric approach where we model all of the red classes as some sort of statistical distribution, blue classes as another distribution, and see what's the probability of fitting to one distribution or another. Okay, so with something like this approach of the nearest neighbor, this is called the k-nearest neighbor. So it's a very common machine learning classifier. And you might want to think about, where would I not want to use that? Is there a reason why I wouldn't want to use this, or I wanted to use a different approach? Well, in this case, we have a lot of neighbors near the voxel that we're trying to classify, right? But if we had one that was way out, not close to our other neighbors, the k-nearest neighbor might have some problems here, because if you have sparse training data, the closest label within your feature space might really have nothing to do with that position, that intensity value. So in that case, some of these other methods might be more appropriate. So here's an example of several different classifiers. I'm not going to go through them in detail, but this is sort of a visualization of the same type of idea, where you have training data, which are in dark red here and dark blue, and then you have points that you're trying to classify, which are in orange or light blue. And we apply different types of classifiers and see what their decision boundaries look like. So you can see that with different assumptions that are made by these classifiers, here, this is the same type of classifier as a support vector machine, but if you use a linear kernel versus a radial basis function kernel, you can get very different decision boundaries. So these different classifiers, there's some rules of thumb that could be used to help you decide on which ones to use, but lots of times people just like to try different ones. They're actually easy enough to use that we can try different classifiers and see which ones might perform best on your data. So another type of classifier are artificial neural networks. So these are parallel networks of processing elements or nodes that simulate biological learning. So these are sort of the craze that's sweeping the nation right now. It's based on neural networks. So if you've heard of deep learning, that's based on these neural networks. And here's an example of a very simple neural network. So we just have 4 different processing elements and 2 layers. Okay, so you take your input, okay, and we're going to pass them through these nodes with different weights. So in this case, we're just perhaps multiplying the input with some weight, and then getting these outputs. And then these weights are actually have to be estimated using your training data. So we're going to get weights estimated by your training data, and once we have the weights, we can take new data and send it through the neural network and get the result that you're hoping to get. So what are the disadvantages of these neural networks? Well, they can be highly effective, but they're mathematically vague. We don't really know what they are doing when they're doing this estimation of these different weights. We don't have sort of an optimization criteria that we understand that's being minimized, this cost is being minimized. It's really just sort of estimating a bunch of weights and hoping that you get the right results. Kind of a black box. So what are the advantages, though, is that because of this interconnected structure, so if you can imagine now, we're going to send a whole image through here with all these connections in between the voxels and all these different weights and computations taking place, that we're actually going to end up, when we're estimating these weights, we're going to extract important features between these neighboring voxels, or not even neighboring voxels, but any voxels within the image. So this is this idea of not needing to choose your features to perform your classifier. The deep network or the neural network will choose them for you. And then another important property is that they can be very efficiently implemented on parallel computers. So when you hear the term deep learning, it's simply a big neural network. I mean, I wouldn't call this a a deep learning network. It's a 2-layer network, but if it has lots of layers, then you would call it deep learning. And it's shown to be highly effective in a number of applications and increasingly being used for segmentation. And the main disadvantage is that because you have this large network and you're estimating all these weights, that you typically require a large training data set. So what I'm showing here is just an example of a deep learning network called DeepMedic that's been used for a number of different segmentation applications. I won't go through it in detail, but there's actually a lot of resources available that can be downloaded. You can download this deep network. There's a couple other ones here, and there's an NIH mailing list on deep learning that, if you're interested, is worth subscribing to because they have frequent talks on deep learning. Okay, so this is not a segmentation method that I'm talking about here. It's an important step for a type of segmentation method called deformal registration. So if you're familiar with fMRI, we know that it's common to normalize these images in some sort of common space. And to do that, it requires a deformal registration. So this is just an illustration of that here. We have an image that we're trying to deform to a target image. And you can see what happens as we apply one of these deformal registration algorithms. It basically contorts the image until it fits into the target. And if you put this original image into some sort of grid, you can see what's happening to the image by looking at the deformation of that grid shown here. So you can think now that if I actually have this ability to warp some image into another image, if I have a bunch of training data sets where I know where the labels are, can't I just take those labels and warp it onto your image that you're trying to segment? And the answer is, for the most part, yes, you can do that. And these are a class of methods called atlas-guided approaches. And more specifically, what's most commonly used are multi-atlas label fusion methods. So we can do this deformable registration with a single atlas, but it's often insufficient because of large differences in the anatomy that can't be accommodated by the registration. So in this example, I'm showing what happens if you try to perform a segmentation based on a single atlas result versus fusing the results of 30 different atlases. So you can see that this is actually kind of an odd-looking brain. It's not that the ventricles are larger than normal. It's kind of oblong-shaped. And if you just apply that one atlas, you don't really get a very good segmentation. This animation is actually showing the evolution of the segmentation as you increase the atlases from 1 to 30. So you can see that here you start off with a very poor segmentation, but quickly it converges to a much better result. And then as you're getting to higher numbers of atlases, you get some refinement in the cortical labels, but the essential part of the image is actually labeled pretty well. So fusing those atlases together, there's techniques called label fusion. And so one of the most simple approaches is to do a majority voting. So if we're warping a bunch of atlases onto this image, and we now have a bunch of different labels defined by the different atlases, which label gets the most votes? And that's the majority vote, so we just assign it to that label. And there are more sophisticated methods there. And then one thing related to deformal registration that's worth discussing is voxel-based morphometry. So the idea is that we want to look at group differences by deforming images into a template space and seeing differences in how those deformations take place. So in this example here, we have a not so happy face. I guess it's just a apathetic face being deformed into a happy face. And if you look at this grid being deformed, you can see that some voxels or some pixels are being compressed and some are being expanded. And the idea is to look at that, and you can compute the amount of compression or expansion with a quantity called a Jacobian. where it's bright where there's volume expansion and dark where there's compression. And now if we're looking at trying to do a group analysis, we do that with a bunch of different subjects. And we now have bright areas where there's lots of volume expansion, and dark areas in areas where compression, and we can make the analogy to functional activations. So we can have higher functional activations here, lower activations here, and we can do almost the same analysis that one performs for fMRI. So the steps are really, we do need a segmentation as an initial step, and then we can compute these modulated images, which are modulated by the Jacobian. And then, as you can see, these maps are very similar to what you get at the end of the fMRI analysis, where this is showing, okay, so these ventricles actually are expanded quite a bit in MS patients, and the thalamus is shrinking. Okay, so I'm going to quickly talk about validation. So there's when we have these segmentation algorithms, how do we know how well they're working? So most commonly, we're going to compare it to the manual delineations, but it's also possible to use a simulation, a computational phantom or a physical phantom where you know the geometry, and we compare it to what you obtain with the segmentation algorithm. And then there's a wide variety of figures of merit that I'll mention quickly that you can use that can be based on region information, such as the number of mixed classified pixels, distance between the true boundary and the estimated boundary. A metric that's not commonly used is reproducibility. I mean, we use this a lot for testing other kinds of algorithms, but because most of these segmentation algorithms are deterministic, reproducibility is 100%. So unless you can scan the same person several times, we don't. That's one way to look at reproducibility. But if you just run this algorithm again and again, you won't get a different result. So this is showing here an example of several different lesion segmentation algorithms that we actually compared in a challenge that we organized. And you can see it's actually not that easy to compare these algorithms. They look very similar. And so if you want to make a comparison, you need to think about, well, what are the important outcome measures of our application? In this case, we were just sort of doing a overall comparison of, there wasn't a specific scientific question, but in practice, you would actually want to really consider what your application is. Here's a list of different validation metrics we use in this particular challenge. You can see there was a large number of different ones. And what was kind of interesting here was we actually compared the importance of the different types of metrics to a neuroradiologist's subjective rating of how good a segmentation was. Okay, so he looked at the different segmentations and said, okay, this one's good, this one's okay, this one's not so good. And we looked at, using machine learning, we looked at which metrics relating to his rating to these metrics, what were the important ones. So it was actually interesting to see that this false positive, lesion false positive, ended up being a very important one. And you can think about if a radiologist is looking at a segmentation and he sees a false positive, that stands out, right? He says, oh, he's finding a lesion where there's no lesion, this will stand out in his rating. True positives really didn't factor that much in his decision making, and part of the reason for that was that all of the algorithms actually were quite sensitive to the lesions. So generally they found the lesions, but the differences between the methods were in how many false positives they had. Okay, so that's false positives where we found a lesion where there shouldn't be. I'm going to skip this portion. I have a bunch of slides on a sort of example application, but I'm running out of time. So I wanted to leave some time for questions, but basically, in the end, the main thought is that in actual practice, when you're applying segmentation, it's not going to be a simple application of one of these algorithms that I just described. It's going to be usually a combination of different approaches that will allow you to get the best result. Okay, so in this case, we used many different types of algorithms to first get a skull stripping, lesion segmentation, and then eventually a whole brain segmentation and a cortical segmentation. And then a specialized approach for just getting the thalamus because we wanted to use the fusion imaging for getting the thalamus. So there's many different components. If you think about algorithms such as FreeSurfer, it also has many different components. So quickly with future directions, one of the areas which are still under a lot of development is how to deal with pathology. You know, it's kind of crazy to think of the fact that most of the time we're segmenting healthy brains when, you know, it's not really that important to segment a healthy brain. They're already healthy. You know, you really want to understand these disease processes. So lesions, but this can be difficult because lesions can have a very heterogeneous appearance. So this is a TBI patient at 3 different time points after their injury. So this is the first time point, second time point, third time point, and you can see that actually the lesion, this hemorrhage here, actually changes appearance depending on when you acquired that image. And this is a bunch of tumor patients, and you can also see that there's great heterogeneity in the appearance of the lesion. And it's not just lesions that give problems. It can be sort of extreme variations of anatomy. So for example, here is a person with normal pressure hydrocephalus. And if you apply FreeSurfer, standard FreeSurfer, you get this result. If you try to tweak FreeSurfer, change the different parts and just try to get the best result possible, you still get a bad result. And that's because FreeSurfer is using this probabilistic atlas where it just doesn't believe that the ventricles can ever be this close to the cortex. Right, so it's never going to be able to find a good segmentation of these ventricles. All right, and then one final aspect is that if you acquire MR images with different protocols from the same patient, and you apply the segmentation algorithms to those different MP rate and SPGR in this case, you'll get different results, even though the underlying anatomy is the same. So this is a disappointing property of segmentation algorithms, but they are dependent on the protocol that you use. So this is another area of interest in the future. So just some conclusions. I won't go through them, so I can leave time questions. Structural MRI and fMRI should be friends. That's true of the data and the researchers. So befriend your structural MRI researchers. And hopefully I gave a sense of the large number of approaches and publicly available tools that are available for segmentation. And I'll stop there. Thank you. Any questions? Right, so the question was, in the competition that we organized, how did we actually decide who the final winner was? Is that right? Okay. Oh, okay, so this is sort of a meta-analysis approach, fusing different segmentations. We actually, I'm not sure we actually investigated that in the end, but it wasn't actually necessary because the level of performance with some of the approaches were actually as good as the difference between 2 manual raters. So the standard of our competition was, can you be as accurate as the difference between 2 manual raters? And it turns out that lesion segmentation is very hard, so 2 manual raters are not very close to one another. So, so that was achieved, certainly by some of the algorithms. They were able to get as close to the segmentations as 2 manual raters. Yes, there are a number of tools that actually deal with inhomogeneities and subcortical segmentations. FreeSurfer is not that bad, but there are probably more sophisticated ones out there. So if you want to go state of the art, I would say some of these deep learning tools that I mentioned, I have some references to DeepMedic and one called DeepNat. DeepNat, I think specifically, is a whole brain segmentation approach, deep learning approach, which probably sort of represents the state of the art in terms of getting whole brain segmentation. In terms of inhomogeneity correction, typically people are applying a pre-processing step to remove that. And that works quite well at 3 Tesla. It doesn't work well at 7 Tesla. It's not really sufficient at 7 Tesla, but for 3 Tesla images, there's an algorithm called N4ITK, which works quite well. Mm-hmm.
Yeah, know anything about neural net
I'm not familiar with Norfolk. Do you know which group it came out of?
It's out of South Korea.
Oh, okay. So yeah. So I haven't heard of that, but yeah, FreeSurfer is one of the slower algorithms out there, and certainly if you have a parallel computer, these deep learning algorithms can perform quite efficiently. So I guess it also depends. I don't know what the details of the algorithm are, what structures it was trying to find, but part of the process that makes FreeSurfer slow is not just the segmentation, but the cortical reconstruction process. And so if it's just finding the whole brain segmentation, then it's sort of missing that last step that FreeSurfer does, which takes a lot of the time. Any other questions? Yeah. Hmm? Okay, so the question was, what are the differences between the atlases used in, for example, the multi-atlas label fusion? So as I might have mentioned, I believe I mentioned that training data is hard to come by. So the 30 atlases are, in this case, were 30 healthy brains from the OASIS data set. And these were the free ones that are available in the Vanderbilt challenge, that were available from the Vanderbilt challenge. They're healthy brains, random people. I don't know what the exact exclusion and inclusion criteria were, but they were free for the picking, so that's what we use in this case. So if you get the training data for neuromorphometrics, they also have ADNI data that's hand segmented as well. Any other questions? All right, thank you.